All right, well, thanks everyone for being here again today. I'm going to talk to you about what happens when you already have your BAM that you processed through the best practices as we described yesterday, and you want to try to get some meaningful information out of your BAM, which for the most part means how to get from a BAM to a list of genomic variants. So here is your BAM. You know, we described the process here yesterday. And now that you have your process BAMs for one or for many samples, I'm going to describe the process of variant calling. So the variant calling process is the process that takes several of your samples, the BAMs of several of your samples, and then it converts to raw variants. It could be SNPs, it could be indels. There is, of course, also the case of structural variation, which is not covered, but I think that we're going to have at some point a breakout session on genome strip at some point later in the day to describe how to sort of leverage this to also discover structural variants. But that's not part of the focus in this session. So once you have your variants, Ryan is going to describe later on what happens when you actually want to filter those variants to sort of weed out your false positives, to diminish the artifacts with the process of variant recalibration. And then later on in the day, we're going to cover what happens with genotype refinement and how do you actually evaluate the variant. So that's more or less a roadmap for today. It's this right part of the figure, and I'm going to start describing the process of variant calling. So the purpose of variant calling is mainly to discover in a formal, systematic way the genomic variants that are hidden in your data. Now, the problem in general is that it's not as simple as just, you know, looking over your reads and then just like, essentially saying that there is a variant every time there's a mismatch with the reference. Why is this? Well, the reads are not perfect, there's a bunch of noise, and you want to sort of tease out the signal from the noise. So this is actually a case where it's in some genomic region which is in general clean, but here, for example, you see something that may be a SNP, but who knows, right? I mean, like, you know, there are some mismatches here in the green, so there are these bases in green, but there are also some insertions, there are a couple of deletions. This is actually not homopolymer, so who knows? I mean, this kind of looks suspicious, and you know, we sort of need like a formal way of actually teasing out a true genomic variant from just random sequencing noise. So as I was saying, you know, what we're dealing with here is essentially a large-scale Bayesian inference problem that is going to empower us to distinguish a generic variant from random machine noise. And in general, we take two approaches. The first approach, which is sort of like historically the most important and the one that we've been working on for a longer time, is with the GATK tool Unified Genotyper, which I'm going to describe in a little bit, which essentially it walks position by position on the genome and tries to find out whether there is variation there. So going back to like this picture here, what Unified Genotyper does is that, you know, at this particular position, it looks at all of the bases aligning to that position to see if there is some variation. Regardless of whether it finds something or not, it walks to the next position and it repeats the decision process again and again and again. So this sort of position-independent approach is very fast. We sort of know how this works, and we have a lot of experience sort of like filtering out the variation from the true noise, and it's what we use actually in the production pipeline here at the BroadE, and it in general works very well, but it has several shortcomings, and especially when we deal with more complicated variants, when we deal with indels, with complex substitutions, where or with large genomic events that are not so simple as just, you know, a plain SNP, which is in general clean, then we have to take a more evolved, a more computationally evolved approach, and this is what the tool called haplotype color does, which I'm going to describe also a little bit later. And this actually involves looking for regions that may harbor some interesting variation, and at these regions we perform a local de novo assembly of the reads to find out whether there is a variation there or not. So these are the two approaches: unified genotyper, haplotype caller. Unified genotyper, as I was saying, is based on locus independent. It has the advantage currently that it accepts aneuploidy. So if people are dealing with like haploid genes, haploid genomes, or like pooled experiments or polyploid organisms, it's supported by unified genotyper. Haplotype caller at this point, it doesn't support that, but it's a feature that we will work on in the near future. Unified genotyper also can accept very high numbers of samples. So with, in conjunction with reduced reads, we have been able to push it to like maybe 60,000 samples, something like that, simultaneously. And so it really scales very well with number of samples. Haplotype caller, it's not there yet. I mean, eventually it will be, but it's still a work in progress. The advantage that haplotype caller currently has is that it's more accurate, especially when you're dealing with indels or with, for example, large events. So you know, eventually haplotype caller will be just the tool for variant calling, but that's going to still take some months to get there. So I'm going to describe now more or less the basic principles of unified genotyper so that people can sort of know what more or less goes under the hood when you run it. The way unified genotyper does is that it sort of independently considers each locus, as I was saying, and it also independently considers whether at a particular locus there is a SNP or an indel or both. So basically, the basic workflow is that for each of the samples, for each of the possible genotype present in a locus, it will determine the likelihoods of the data of the genotypes. It computes an allele frequency distribution, and I'm going to go into detail of this in a little bit. And if there is sufficient statistical evidence that there is there is some variation there, it's going to emit a variant call. And the output of both unified genotyper and haplotype caller is a VCF, a file format, a text file format that we described yesterday, and we're going to see also how this looks like in a little bit. So SNP and indel calling, for people who are interested in the math, is basically a large-scale Bayesian modeling problem where we are interested in essentially the genotypes given the data. It turns out that actually it's easier to compute the data given a genotype. So by Bayes' rule, you can sort of do this inversion, and then if we're doing like a diploid organism, then essentially what we have to do is that for each possible pair of genotypes, then you have to compute sort of the likelihood of your data given each of these possible genotypes, and then you combine them. And then we use an assumption that all of the reads are independent, so that for each of the reads, we just sort of like multiply the probabilities or add the log probabilities. So this actually in the end gives you the likelihood, essentially your genotype likelihoods for each of the possible genotypes. And it's, you know, for the SNP case, it's based on a very simple model where the likelihood of the data that you see at a read given a particular base is just either the base quality in linear space or y minus that, depending on if there's a mismatch or not. Question?
How do you compute your prior probability?
So the prior probability by default is just the prior probability of your having a particular variant at a site, given an infinite sites model, you know, from population genetics. So basically, you know, the prior probability of having like an AC of n is like n over m, where m is your total number of chromosomes. And I probably have, I probably have it in the slide somewhere here. But any other question? Okay. So there are several tricks here. One of the tricks is that we only actually use, when we're computing the SNP likelihoods, we're only using sort of like the good bases. The good bases meaning the bases that satisfy a certain quality threshold, and we're also only using the good reads, meaning that we only use reads that satisfy, for example, a minimum mapping quality and several other filters. But in the end, you get like these clean genotype likelihoods, and then those are sort of passed forward to the algorithm. The indel genotype likelihoods are a little bit more complicated. Basically, what Unified Genotyper does is that it's sort of restricted to the indels or the gaps, if we say, present on the reads. So if you recall from yesterday, the cigar string was the, the part in the BAM that sort of described the alignment of a read to a reference. So Unified Genotyper relies on those gaps, the insertions or the deletions produced by the alignment process to actually discover candidate haplotypes or candidate alleles for the indels. So once it has these candidate alleles, it forms haplotypes, and then it computes the likelihoods, the genotype likelihoods, as using a pair HMM model. So this is a standard HMM with affine gap penalties, and if people are interested, we can go later on over the math on the details of how this is done. But this is essentially sort of the way things work with indels. Now, the way that then multisample calling works for both SNPs and indels is that once you have your likelihoods for each of the individual, be it likelihoods for the SNPs or for the indels, we compute the joint estimate of the allele frequency, or rather the allele count across all of the samples. So the allele frequency spectrum, which is defined as just the probability that a particular allele count is equal to I given the data, is computed mathematically by this recursive formula. If people are interested, there's a link here that a paper that Henkley wrote about how to do this. And then once you have that, if we find that the statistical evidence is strong enough for a variant, we say that this variant is present. We assign the genotypes to each sample given based on the likelihoods. So, the most likely genotype is just assigned to each of the individual, and that's how actually a call is produced. So, in general, it's a very, very simple workflow. I mean, you just take a BAM, you run Unified Genotyper, and then get your raw VCF file. So, it doesn't get, simpler than this. Obviously, there are many options that you can actually put into the Unified Genotyper. But in general, the command is very, very simple. I mean, you just, specify Unified Genotyper and your reference. You want an input BAM and an output VCF. And then you have all of these parameters and thresholds that you can tune based on your application. So for example, here, this is sort of like the threshold of confidence that you want to establish for emitting a call. And this is in Fred scale. So for example, a threshold of 10 means that you want to emit every single variant that has a probability of at least 90% of being present. So there are other options to actually run the tool. For example, there's a very popular option called genotype given alleles, in which instead of letting the tool discover the alleles for you, you say, okay, I have this set of candidate alleles. It can be SNPs or it can be indels. Just tell me if these candidate SNPs are in the data, and give me, some information on the statistical evidence for that. This is obviously very, very important for sort of like validation and replication studies where you say, okay, I'm going to sequence this particular region and I'm just interested in whether this mutation is there or not. Just tell me, is it there or not? I don't care whether you discover things around it. I just, is it there or not? So this is the purpose of genotype given alleles, and there are a whole bunch of options to operate this. So once you're done with this, you have a raw VCF file that you can sort of pass downstream for processing. Now, something important to consider is that Unified Genotyper sort of by default tends to overcall. So we sort of, the philosophy is that, we try to call every possible variant given a particular threshold, and then we rely on post-filtering to sort of whittle down the false positives. So, by default, in the sensitivity versus the specificity trade-off, in the raw calling, we tend to be more sensitive, and then, we rely on a VQSR to sort of whittle down the false positives. So, getting up to, the second tool, which is haplotype color. Haplotype color, as I was saying, is a little bit more, is quite more advanced and more complicated, and in the end, it will replace unified genotyper because it's more accurate. It produces better results, especially for indels. And the way haplotype color works is a little bit more complicated. So the way it first tries to get candidate alleles, be it SNPs or indels or even like small structural variants, is that it takes, a region of the genome and it assesses first whether what we call a region is active or not. So basically, it assesses some sort of probability that there is some underlying genomic event that we may find interesting. So, it's a whole bunch of heuristics that we use for this. And once we determine that a region is active, which means that a region has the potential of having some variation, we do a local assembly of the region. So essentially we take all of the reads, we assemble the reads using a DeBruyne assembly or other assembly approaches, a little bit more advanced. And then once we have the assembly, the paths in the graph are actually the potential haplotypes that need to be evaluated. So I don't know to what extent people are familiar with de novo assembly, but for example, the typical de novo assembly of of an initial whole genome, the goal of that is actually to produce most of the time like a single haplotype that represents like your organism, or maybe a couple of haplotypes or a couple of paths within a single haplotype. And this actually is a little bit more general than that, in that we're interested in like all of the possible paths to the graph because those are potential haplotypes. So once we have this list of potential haplotypes in a region, then we use the same idea, the same philosophy that unified genotyper did for the case of indels, where we would sort of try to compute the probability of a read having come from that particular haplotype. So this is actually what our pair HMM model does. So it just tells us, given a particular haplotype, or given a better said, a particular set of haplotypes, what's the relative likelihood that each of the read came from each of the haplotypes. So then there are also other heuristics sort of to limit the number of haplotypes, because otherwise this sort of has the potential of exploding. But then once you have these likelihoods, the process is very similar to what unified genotyper has. And we use the same math to compute the allele frequency distribution. And then once we have the allele frequency distribution, and if we determine that there's actually a good chance that there's a variant, then we emit a variant into a VCF. Yes? How do we define a region? So a region is basically, when we start looking at the reads, and when we find that there are, for example, many mismatches to the reference at a particular position, or when there is like a large number of soft clips, which are places that the aligner said, okay, I'm going to just like cut off the read because there are a lot of mismatches in a read with respect to a reference. We have these heuristics essentially that when we count these number of like mismatches and when they exceed a certain threshold, then we say, within a particular region, then we say, and it's a window, it's sort of like a sliding window. Then we say, okay, this region is a good candidate for having some variation, and then we sort of like process it downstream in this way. Yeah. Mm-hmm. Okay, so this is more or less in detail how it works. This graph is a little bit obsolete because we came up with this new way of assembling reads, but in general, it sort of gives you the gist of what still goes on. Basically, if you have a pile of reads here, a traditional way of doing, for example, assembly is called an overlap graph, in which, for each of the reads, you compute sort of like how much a read overlaps with the others. An alternative approach is this DeBruyne graph, and there's a lot of literature that you can consult on how this is done. And most of the newer assemblers work, or actually I shouldn't say most, many of the newer assemblers work based on this principle. And essentially, the way it works is that for every k-mer seen in the reads, that corresponds to a particular node in a graph, and then you have an edge if there's a k-mer that connects, or if there's a read actually that connects to k-mers. So for example, here, when you start with read 1, you see GAC, and if you assume that your k-mer size is 3, which is too small for practical purposes, but it's good for illustration purposes. If you first see the k-mer GAC, you add a GAC to the node, and then you walk to the next possible k-mer, you slide to ACC. Since you haven't seen ACC, you add it to the node, to the graph, sorry, and then you add then CCT, and then CTA, and then TAC, and then if you, for example, here when you go to the next read, if it turns out that you saw already ACC, you don't add it again. I mean, you just add a weight to the edge to make sure that it that gets captured. And then in the end, you have this graph with branching points representing possible variation. And then if we sort of proceeded further with the graph, you would see that, for example, for every SNP or every possible SNP that you have, there would be like sort of 2 possible paths in the graph. So this is what we call a bubble. And then you can decide whether you want to actually produce haplotypes from these bubbles or from these alternative paths in the graph, or if, just to keep the complexity to a particular manageable level, you may want to decide, okay, bubbles that have only like a very low weight, I'm just going to trim them out. So there's a bunch of heuristics to actually limit the construction, the complexity of the graph construction, because these graphs, when you have like real data, can get very, very complicated. So you want to like make sure that you keep your computational cost reasonable. But we do that in the haplotype color, and then we have managed to have the haplotype color run maybe 2 to 3 times lower than unified genotyper, but it's getting there. So and there are some data sets in which it's actually even faster now. So in the end, soon it will be as fast and as good as unified genotyper in the overall case. So once you have these haplotypes that you read from the graph, you do exactly the same evaluation that you did with unified genotyper for the indel case, except that here you do it for like a whole haplotype, even if it's a SNP. You use the same generative model for the HMM with affine gap penalties. You can actually derive the gap open penalties from the new BQSR or not. And then, the mismatch penalties are just your base qualities. So this is actually very, very similar to the case of unified genotyper, and it actually works very well. So what does this give you? I mean, so you go through these huge numbers of very cool algorithms, but in the end, they cost a lot to actually run. And what does this give you? Well, this is, for example, the read data that the aligner came up with at a particular region. And if you run this with unified genotyper, what would happen? Well, you would run on a side-by-side basis. You would see, well, this is actually a good SNP because there's a lot of Cs, very clean Cs. So I might say, okay, this is a heterozygous SNP. The insertion, there's some insertion here. I may want to call that again, and then there's another T insertion, and then some deletion. But then this is actually not the most parsimonious representation of the data. What's happening here is that there's a large deletion underneath. And it just happens that because of the tandem structure of this deletion, that the mapper wasn't able to actually open this big gap, and this is probably like a 25 base per deletion or something like that. You know, the mapper, BWA or any other tool, just wasn't able to open this gap, and then, you know, it tried to actually do the best it could, you know, and it just placed these reads with these mismatches. And there are other cases that we see which are very common, in which, you know, when you have like these sort of large deletion, what happens is that, you know, the mapper is going to place, you know, the bases at one edge of the deletion, and then it's not going to know what to do with that, and then it's going to soft clip the rest of the bases. So essentially, you know, it's going to try to throw them away. I mean, luckily, they're not really thrown away. They're just marked as clipped, but, you know, we can recover them. So that's actually a very strong signal that there is an underlying true event there. So, you know, this actually is a huge improvement, but you wouldn't be able to actually see this just with a locus by locus view as unified genotyper, and that's why you need all of these more sophisticated assembly methods to actually get these large deletions. And so with haplotype color, we can get, depending on your data, you know, we can reliably get up to like maybe 70 or 80 base pairs. It depends on the data and the length of the read. I mean, but, you know, in general, we do a very good job in calling like large events, both insertions and deletions now with haplotype color. That with unified genotyper, it just wasn't possible, and this sort of filled the data with artifacts. So there's another bonus here in that you can actually get phasing for free, because, you know, since we're calling haplotypes instead of just locus by locus, you can have actually the phase information. So for example, here, this is actually a multinucleotide polymorphism. So this is one particular gene where, if for example, if you run unified genotyper, you would get like 2 records, right? I mean, 2 SNPs, which is useful and it's correct, but it wouldn't give you the whole picture, because especially, for example, in a clinical setting, when you actually do variant interpretation, the presence of one SNP in a coding variant is very, very different, obviously, from the presence of 2 SNPs. So, for example, this one in the end, I mean, I don't know my genetic code by heart, but, you know, it could very well be that this second SNP could be a synonymous mutation, right? And then you know, if looked at in isolation, you would say, oh, it's synonymous. You know, maybe it's not effectual. But, you know, if you always see it in phase with this first SNP, so it's a compound head or a multinucleotide polymorphism, then this is a completely different picture, right? And you know, also from a clinical interpretation standpoint, I mean, this is very, very different, and this is very important information to have. So what you want to have is sort of a record that these 2 things go always together, and this is what we get when we're calling these haplotypes instead of like a locus by locus view. So there's another advantage to this in that you have like a more consistent representation, especially of complex events. So this is an example where you have one insertion and then a SNP, and then an A, and then another insertion. So, you know, a naive caller would say, okay, there's an insertion here. There's actually a multinucleotide, a multi-allelic SNP here, because, you know, the reference is T, but then, you know, some reads have C and some reads have an A, and then you have a separate insertion here. So, you know, this is probably from a you know, technical perspective correct, but it actually turns out that the underlying bases in the BAM are the same. So this event in the top is exactly the same event in the bottom. What happened here? Well, it happened that because of the vagaries of the way the mapper, you know, BWA or any other tool, because of the way they actually produce and open the gaps, they actually produce one representation for a set of reads and another representation for a set of another set of reads in the same locus, in the same sample. So this may have to do with the parameters that they use, for example, for Smith-Waterman, you know, when actually doing sort of like the placement of the read. But in general, this is bad, right? I mean, because, you know, this doesn't give you like a good representation of what's going on there, you know. So what really happens is that, you know, the bases TT are going to TAC, and this is like one single event, and the assembler, I mean, for the assembler, this is a very, very simple case, but you know, if you're limited by this locus by locus view of the world, then you're just not able to see it. So this is a very cool thing that haplotype color can do now, and in the end, you know, this only helps you with the accuracy of your calls. So in terms of the protocol, it's very, very simple. It's very similar to Unified Genotyper. Most of the arguments that, of Unified Genotyper that you've grown to love and to cherish will probably work in haplotype color. So all of your thresholds, or most of your thresholds may work. Probably the only big difference is that if you don't specify anything to Unified Genotyper, it's only going to do SNPs. So you have to tell Unified Genotyper if you want to do indels or if you have to do both. This does everything by default. But otherwise, it's very, very simple. So you have like, you know, the same set of parameters, mostly the same parameters as Unified Genotyper, and some extra parameters related to, for example, the graph. You know, I was mentioning before that when actually we build a graph and we build haplotypes from a graph, we may want to sort of limit the number of possible paths through an assembly graph. And we do that by pruning the graph before actually doing the full HMM evaluation. So we have these arguments that control the pruning of the graph. And you know, we've set up the defaults actually to be reasonable in most cases, but people may want to play with them, you know, if their data is, you know, too shallow or too deep, or if you have like too many samples. So, you know, you're very welcome to actually experiment with this and to see how this actually affects the performance. So, yes?
Sorry, does it work with the reduced spam?
Yes, yes. Mm-hmm. Yeah. It doesn't work, however, yet with haploid or multiple ploidy genomes. That's the current implementation restriction. Mm-hmm. Any other question? Yes? Yes, so the question is whether it's possible to actually get the output BAM file. Yes, there is an option actually to essentially have an output BAM file with the reads aligned to the haplotypes that it's considering. The caveat of that is that it's very expensive computationally. So, you know, it's good for like, you know, small localized experiments, but you don't want to do it like in the whole genome because it's going to take forever and you're never going to finish. Mm-hmm. Any other questions? Okay, so in the end, you have this raw VCF file, and then the question that, you know, people naturally have once they run their color and they have this VCF, which is just this, you know, big table of mutation, is, you know, is this any good? You know, am I doing a good job in getting the variants that I have? And you know, obviously, there are sort of two dimensions. You know, the sensitivity dimension in that, you know, the question is, am I getting everything that I'm supposed to be getting? And the specificity dimension in that, you know, is all of the stuff here real, or is there junk here? And you know, the answer is that you know, there will be real stuff, but there will also be junk. You know, we cannot avoid the problem of false positives in general, but we actually have very good and very powerful tools to actually in the downstream to actually assess the quality of a variant call set and to filter out the false positives to actually get yourself a very good set of variants. So this is where we are. We're here. We called variants. We got our list of raw variants, SNPs and indels, and the next step is going to be then variant recalibration, or in general, you know, how to filter out the false positives in your data. So I encourage you to actually go to our documentation. Take a look at the best practices. You know, the best practices are a little bit different if you have, for example, you know, whole exome or low-pass whole genome, or, you know, deep whole genome. You know, the thresholds are different. The recommendations are a little bit different. You know, you might have a case where you have like too few samples, in which case you may want to like sort of spike in more samples from other public data sets to actually get, you know, get better results. So, you know, again, you know, go through the documentation, take a look at the tutorials and everything that we have there. and welcome to the brave world of variant calling. Before I turn over to Ryan, are there any other questions related to variant calling? Yes.
So you mentioned haplotype genotype haplotype color is almost there.
Yes.
To where, so at this point, would you say for SNP calling still use unified genotyper, or is haplotype color good enough for SNPs and it's just not there on the other aspects?
So the question, just for the record and for everybody to hear, is, is right now haplotype color good enough for SNPs? I would say for SNPs, then yes. It may lack a little bit of sensitivity, especially, for example, if you have like low pass data. And the reason is one of computational complexity, because like if you have like a lot of samples, 1,000 samples with like 4 or 5x, the number of paths in your graph is just going to explode. And so you're going to have to do a very aggressive trimming to actually keep your computational costs sort of in line. And then by doing that trimming, you may actually reduce your sensitivity, especially to low frequency variants. So this is a trade-off that we have to do. For exomes, I think we're almost there. I mean, the only, as far as I can remember, the only trouble with exomes is just a plain computational cost. But in terms of,
if you have many samples, probably.
Yeah. So if you have like 10,000 samples, you may still want to use unified genotyper to call your exomes, not because your results are not going to be good, but just because it's going to take longer. And you may or may not have the available farm resources to do that.
And also taking into account that haplotype color is still undeveloped, so it changed more rapidly than unified genotyper. This is the main two reasons that we still use the unified genotyper in the production.
Yes.
Do you, for example, use RNA-seq data?
We don't have experience with RNA-seq data. I mean, the problem with RNA-seq data, in principle, it should work in the same way, but we need to do a little bit more work in the infrastructure side to support, like, for example, these the gapped alignments and all that stuff. So, there's some infrastructure involved in that we still need to do. Once we do that, in principle, you should be able to do it to actually detect SNPs there. But for now, we don't really support that.
So how would that be different than exome alignments?
Because with exome alignments, what you're doing is actually you're having baits that you're capturing your data with, and then your reads are once you have a read, it just comes from a particular place in the genome, right? Since the RNA-seq data comes after splicing, you may have to like open up gaps in the introns, and those gaps are things that we're still having trouble with in the engine to deal with.
The indel calling between unified genotype and haplotype color, similar statistical model or
the statistical model for the indel calling in haplotype color and unified genotyper is the same. So the HMM formulation is exactly the same. The main difference is the way in which we discover candidate haplotypes between one and the other. And as I was saying, the haplotype color is using this assembly approach, local assembly approach, which is much more powerful. So it allows you to discover, like, better, more realistically, the events.
Is there any comparison between SAMtools and pileup calling indel versus genotyper calling indel? Is there any performance comparison?
So the question is whether there is any performance comparison between SAMtools and pileup and the GATK unified genotyper. There have been several papers actually sort of comparing the performance of several colors. I don't specifically recall if SAMtools was part of these evaluations, but there have been published papers in the literature sort of comparing GATK against X, Y, and Z, and in general, the results are that everybody is more or less very good with SNPs, but indels are very, very discordant among algorithms, and it's still very hard to assess, if one is particularly better than the other. Mm-hmm. Yes.
Colors take strand information into account?
Well, the question is whether the colors take read strand information into account. They don't to actually produce like raw calls, but it's an integral part of the filtering process and of the of VQSR that Ryan is going to talk about. So you're going to see that, you know, one of the main covariates that we use for filtering is actually the strand bias information. So in that sense, it is used actually to detect artifacts. Yes?
How do these colors handle, like we have cancer and we have subclones that are low allele frequency, like 10 to 15%?
Okay.
Not the expected heterozygous.
Right. So the question is, how do the colors actually handle, for example, cancer cases where you have like some variants with subclonal frequency of 5% or 10% or something? So the short answer is we don't, and the best recommendation for that case is actually to go use MuTect. MuTect is based on the GATK. It's sort of using the GATK framework, but the cancer group here has like built all of these very specific ways to actually deal with cancer data, and it's sort of like the best practice to actually do that. I mean, in principle, you might use unified genotyper with like a higher ploidy setting to actually get those variants, but your results might not be good because you may actually have like artifacts confounded with like these subclonal variants. So the best recommendation in that case is to use MuTect. Yes?
Are there any special considerations or differences in performance regarding reads coming from different sequences with different read lengths, error rates, things like that?
So the question is whether there are difference in performance and consideration based on essentially the underlying technology, whether it's read length or technology. So yes and no. In terms of like read length, we have handled more or less with the same, with no changes, ever since we had like this Solexa GA reads of like 25 base pairs, was it the first generation, something like that? All the way from 25 base pairs to like 250 in the most recent Illumina runs, and it just works. We do need to be especially careful with technologies like ION or 454 or even PacBio that their main error mode is indels. So the problem with that is that for example, if you want to run haplotype color with those reads, like the number of paths is just huge, right? And you may not be able to do a good job in actually building these haplotypes just because the number of gaps that you're dealing with is just exploding. So we don't have that much experience dealing, for example, with haplotype color with that kind of data, and I think that you probably would have issues. But that's sort of the main consideration. I mean, otherwise, in principle, it should work, but that's an area to consider. Any other question? Okay, so I'm going to turn it over to Ryan, who's going to talk about variant quality score recalibration. Thank you.